Welcome to another episode of Whose Gene Is It Anyway? I am your host, as always, Justin. Welcome to the show. I think it'll be good. It'll be fun. Anyway, welcome. I got a couple little things to touch on before we get into it. We'll draw a topic in just a moment. Today will be another random idea. I actually just went through the hat full of ideas and kind of culled it a little bit. There were some ones that were a little bit repetitive, or were super overlappy with stuff that we'd already done, so I didn't think it would really be interesting. So I've kind of pruned it down to the most interesting ones. So hopefully whatever we draw today will be a lot of fun. A couple in there are a little bit short, so we might do a double episode, might do a single. Not entirely sure yet. Okay, yep, and my phone locks. That's fun. Yeah, I've just got my phone in front of me, just because I've got a couple little announcements I want to go through really quickly. First things first, and this one I think is really important. There's a new video on my second channel, and I would highly recommend that you go check it out. The channel is The Taste Emporium. There should be a link in the description, and if there isn't, I will add one. Either way, it is maybe the prettiest video I've ever made. The item that we made is like aggressively delicious. So if you like food and you like cooking, highly recommend checking it out. Yeah, so that's the first big one. That channel's doing really well for having only created it recently. We're coming up on it actually being monetized, which is cool. And so yeah, that's highly recommend that. Okay, so that's thing number one. Thing number two. So I just want to talk quickly about why I canceled the stream on Monday. As many of you have noticed, the world is kind of on fire. There's a lot of people who are hurting right now, so it didn't feel responsible to do like a goofy, fun stream when that was going on. And I mean, it's still going on, but you know, I do need to get on with it. So yeah, that was Monday. I just kind of couldn't deal. Been trying to avoid social media lately because frankly it is just every time I open it I'm like, oh good, a new horrible fucking thing. Great. So yeah. Yeah, there's a lot of people who need support these days, and so I decided I'd take the day and do that instead of this. So yeah, that was Monday. Thing numero trois. I'm gonna have a really exciting guest next week on the streams. So for the bazillion of you that have asked about THC producing yeast, a friend of mine actually runs a company here in Canada that has managed it. They've produced a strain of yeast. Actually, they've produced many strains of yeast that actually synthesize THC, and so they could grow THC in a vat. Is the plan, and so they've managed it successfully, and now they're at the scaling point. So they will be we'll be doing that stream next Saturday at I think 2 p.m. But I'll post more details on Twitter and all the other places. But yeah, that's gonna be a lot of fun. I've been wanting to have them on the show for a while, so that's gonna be fun. Yeah, highly recommend checking that out when it comes out. Also, Monday will be the next biohack chat with Gabe. So be sure to tune in for that as well. Speaking of which, if you have topics that you would like me and Gabe to cover, you can leave them for me on Twitter or all the various places that you can message me. Feel free to do that. Always looking for things that you want to hear us chat about. Okay, I think that is... and then the last one is what this new donation bar thing is about. So I've been making videos for many years, and I've been doing so on a laptop, which is probably fine, but it's quite dated. So a huge problem when it comes to editing videos is that it's just very slow. So the actual render, it probably doubles or triples the amount of time that I have to spend editing. So I've kind of hit a point where I'm like, you know what, it's the right time. I need to upgrade this because it's getting to the point where I'm like wanting to rip my hair out as I'm trying to edit. So I think it'll lead to better videos and less stress and just easier ability to do things. So over the next little while, we're going to be doing some different fundraisers to buy the various components. I've already picked out all of the stuff for the computer. And of course, if we don't hit our funding goals, like I'm going to just cover this from ad revenue and whatever else. But any amount that I can, help that along the way is greatly appreciated. So for anyone who feels like donating, that's what that's going towards. The CPU that this one is, it's a Ryzen 9. I don't remember the exact thing. I've got a whole list of bits, but it's a beast. I'm making a point of, like, if I'm going to do this, I'm going to make sure that I get a computer that's going to last me the next 5-10 years, like something that could mean I could render 4K and all that kind of stuff. So that's what that's about. Okay, those are all the announcements. Let's get in. Oh, and also just quickly to address this, you don't render by GPU. Almost all editing software, with the exception of one which I don't use, primarily uses CPU power to do the actual, like, thinking. So it's like CPU and RAM. So the build primarily uses those two things. So it's just a shitload of RAM and just a monster of a CPU. I think it's got 12 cores or something ridiculous. But this way, it'll last. It won't get burnt out, and I can just do the thing and get my stuff done. Quick question from Kevin A. Can we get a sample of the milk plasmid if we donate? So the milk plasmid and the egg plasmid, once I know that they're functional, will be made available for those who want it. So not sure of the specifics of that yet. The people who donated to that fundraiser initially will definitely get a sample. I don't know how I'm going to handle further donations, but I will figure that out. We'll cross that bridge when we get there. And speaking of which, the synthesis company has officially started working on printing printing the DNA. So both the egg plasmid and the milk plasmid are actually being made. So that'll be ready in the next couple of months. It takes them some time. It's not a quick process, but they'll get it done, and I will continue to update as I get updates, basically. So yeah, Art Love, 12 cores is cute, not a monster. Well, compared to the like 2 or 4 cores that I'm used to, 12 cores is a lot. And for just one little rendering computer, like, it's not a supercomputer, I realize, but it'll do the job. Anyway, time to pull an idea, and then I gotta update the stream name. So let's pick something good, hopefully. Come on, something good. Please don't be boring. Okay, I can do that. It says make oil with microbes in my absolute shit-tastic handwriting. Also, we had another donation from Mr. Roberts. Thank you very much. Greatly appreciated. Okay, so what do we think? Let's do some. Let's make some oil. I mean, the nice thing is, so this one will probably be pretty quick because we've technically done this already. So I might do a second one, or we might just look at a different oil. I'm not entirely sure yet. Also, there's some other stuff that can be used for this. So I think it will. I think it could be interesting. So yeah, let's get into it. So I'm gonna pop over to. Just gonna make sure that everything's open correctly. Yep, cool. Okay, let's get into it. So I've got the chat open. I'm just gonna push it into a spot where I can see it better. Okay. Also, before we get into it, I'm just gonna change the stream title because I always inevitably forget this. So let's just get to it now. That's not how you spell that. Okay. Okay, so the title has been changed, and so we're ready to get into it. You need to lube up your microbes. Yeah, gotta have well lubed microbes. Okie dokie. So, okay. So I want to let's start off with actually something that we did in a previous video. So let's start with Cheeseberry McPearface, which is my favorite name for a plasmid because, you know, why name things professionally when you can have fun? Okay, so this was a plasmid that we made in an earlier video, and so just a reminder for people if this is your like first time tuning in. A plasmid is a circle of DNA, and these little arrows are basically labels to tell you what this little stretch of DNA looks like. Because if you these two screens are the same. So if I click on this, you can see that this is representative of a long string of DNA letters. So if you were to just look at DNA letters, like they don't mean anything to a human. Like there's no way that you could just like parse this and know like, yes, this makes, you know, silk or whatever. Like it's sort of like a random, like it's a random string of stuff. Even if you like tell it to translate into amino acids, like you can't look at an amino acid string and go, yes, this is insulin. Like that's not how it works. So labels are very important. This is why we put them on. So there's a few different things that we put in here. The ones that we're going to be concerned with today are these four over here. So just to give you a little breakdown of what this plasmid was meant to do, it's called Cheeseberry McPearface because it's meant to run in a species of bacteria called Lactobacillus casei, which is a bacteria that's traditionally found in things like yogurt or cheese. And in this case, it was, if you were to make this plasmid, put it into the bacteria, it would make the bacteria produce all of these new genes. And each gene does a different thing. So this one, for example, this purple one, you can see that it highlights when I mouse over it. So it's linalool synthase. I'm going to click on it again so you can see it. So linalool synthase. So this makes a compound called linalool. So it's a protein, like it's an enzyme. And when this enzyme forms, it grabs certain things that are already present in the cell and sort of shifts it around and turns it into a compound called linalool. Linalool is described as the scent of Fruit Loops. Or it's sweet. It's present in a lot of fruits and flavor, and like fruits, some vegetables, some flowers. It's one of the cocktail of compounds that is traditionally found in things like strawberries or raspberries or whatever else. But yeah, so that's like what one gene does. So it makes this one compound. These other ones, though, are a little more interesting. So this one over here is acetyltransferase, also known as ATF1. So this sticks an acetyl group onto an alcohol, and so I'm going to actually show that to you. ATF1 gene. So there's a particular image that I'm looking for, and hopefully it shows up, but it's not. Banana. This one. This is the image. I've shown this image many times. Okay, why is it so tiny though? Be big. So yeah, BioBits Explorer. This is a really cool thing. Highly suggest checking this out if you get a chance. Ooh, somebody kicked in. Through super chat. Did I miss a super chat? Oh no, it just didn't. It took a second to show up. Okay. J. Lawrence 6809 says, if you're still accepting ideas for whose gene, add an antifreeze protein from something like carrot into tomato plant, or better yet, a tropical plant. Okay. Sure. I'll add that to the thing. I'm gonna take a note of that. Seems easy enough. At the very least, I'll check this out after the stream and make sure that it's viable. Because I always, any ideas that you submit for these streams, I check to make sure that they're viable before they get added to the hat. So, antifreeze protein in other plants. Cool. Well, I'll check that out. Okay, so back to ATF1. ATF1 does this. This is the graphic that I wanted to find. So what it does is it sticks an acetyl group. This is an acetyl group here, and it makes what's called an ester. So see how this oxygen is between the acetyl group and this other hunk? That's called an ester bond. So it takes an acetyl group, it sticks it onto an alcohol. This is an alcohol. In this case, it'll just do it to any alcohol, or not any alcohol, but it'll do it to most alcohols. It's not overly picky. So in this case, you can see that the same enzyme works on all these different alcohols, and depending on which alcohol goes in, you get a different scent compound on the way out. So isoamyl alcohol becomes banana scent, and this is like a well-known thing. You can buy kits that do this on Adgene, which we'll talk about in a minute, which is sort of like a GitHub repo, but for like chunks of DNA. You can actually buy a plasmid that does this. It's called banana latte, because biologists are largely children and love naming things in fun ways. So yeah, if you mix isoamyl alcohol with the bacteria that produce this enzyme, it reeks of bananas. But if you use a different alcohol, like say hexanol, you get pears. So if we go back to the plasmid, all of these things here are the different enzymes in the hexanol synthesis pathway from glucose. So if you feed this bacteria glucose, it'll take that glucose and using these different enzymes will turn it into hexanol. So like I said, the reason this one might be a little short is because hexanol is an oil. I mean, if we look at it, it's a long chain alcohol. So yes, technically it's an alcohol, but hexanol is fairly similar to like diesel. Octanol though would be a better choice. So what we're probably going to end up doing is a very similar thing, but we're going to go for octanol instead, just because that's a little more oily. You got to figure that every time you add another carbon on here, so this is hexanol, it's got 6 carbons. This is octanol, it's got 8 carbons. So every carbon is more energy in the compound, so that when you light it on fire, more energy comes out. So we're probably going to do octanol. We'll then stick the acetyl group on it, and then you get like this pear compound. Mix it with the linalool, you get like this weird mix, in theory, of like pear and fruity and this sort of thing. And ATF, because it's not really picky, there's all sorts of other alcohols that end up as metabolites in bacteria. So it's just going to be acetylating things. So you're going to get this really weird bouquet of different fruity smells. So hence cheeseberry McPearface. That's where that came from. So now that we know the basic idea, we're going to do the same thing. We're going to have to find this pathway, and I already know that there's a paper that exists for this because I've looked at it before. And then we can do octanol synthesis. And if you've got, and so this has actually been a thing. So the reason why I know this is a thing is because I looked this up in advance at one point. And they'd made octanol not to make orange scent, which is what I wanted, but because they wanted to burn it. So if you had a microbe that you could just feed sugar to and it essentially just poops out diesel, like that could be pretty cool. Especially if you're using like waste streams from other processing plants. So like if you were going to take like the waste from, let's say, pasta production, like when you're making pasta, you get this like lake of starches, essentially. Like this is a really common thing. Like the outfeed of just the process of making it, you end up with this like water that's full of starches that's left over from various processing steps. And so if you were to use that as fuel and like feed it to a bacteria that doesn't really care that it's starches, like it'll just eat it, you could turn all of that waste product into diesel, which would be kind of cool. I mean, I say it's kind of cool. I think it's horrifying because I'm not a big fan of burning things just to, run your explosion engines. I'm a big proponent of, if I can hear your vehicle, it's broken. But yeah, so that's the thing. So we're gonna do octanol synthesis, I think, just because I think that's nice and easy, and we can do this. I'm gonna find, I don't know what organism I want to do this in. I'm kind of leaning towards yeast, just because I already have a nice yeast plasmid that we could use for this. This is the yeast plasmid that we can use for this. So we could do that. Not quite sure yet. Yeah. But yeah, so that's the thing. I'm gonna just take 2 seconds to just read some comments because I've been seeing some questions and I wanted to answer some of them. I saw someone, and I don't know if this popped back up again, asking how many of these streams I'm gonna do and if I'm gonna do them even once quarantine is lifted. Quarantine has lifted. Like, my city is open again. But here we are, still streaming. I've come to really enjoy doing these streams, and I'm going to keep doing them. I mean, y'all seem to be enjoying them. Obviously, they don't get the same kind of crazy viewage and like ad revenue as a traditional video, but I mean, it's a nice excuse to kind of hang out with everybody, chat, talk about how to design DNA like this. It's a it's a really fun excuse to practice my genetic design skills, and it's become very very helpful because I'm finding that when I'm actually working on like legitimate projects and not just like fun stupid shit like this, I'm catching stuff that I wouldn't have previously, just because you spend 10,000 hours doing something, you get good at it. Um, I'm finding that I'm catching my mistakes better, and I'm finding errors and stuff better, and I'm designing better DNA, which is frankly important to me. So I think I'm gonna just keep doing it. I think it's fun. Y'all seem to enjoy it. Yeah, it's a great interaction-y thing, and it's not like I'm gonna stop making the regular videos. It's like I'm at this weird point where the regular videos are still going on in the background. I pick really hard projects, so I just haven't been able to like crank out videos because the things that I've chosen are very hard and they're taking time, and there's also the world is on fire, so getting supplies has become very difficult. So I've got a couple big bio projects that I've been working on for like months now, and until my new order of reagents show up, there's nothing I can do. So doing streams is a way to just kind of keep on keeping on and doing things, and also like when we did the egg and milk thing, like that's become actual DNA. Like we did the fundraiser, people chipped in, we got them made. They are being made. So we're actually gonna make some of these things eventually, like the really interesting ones. So yeah, I'm definitely gonna keep streaming for a little while. Okay. Somebody asked, "What is your day job?" This is my day job. YouTube. This is all I do. Okay. Somebody asked, "Can we create CO2 consuming organisms that make O2?" They're called plants. They exist. Okay. Okay, I saw one other question. I just want to see if I can find it really quickly because it was interesting. And if I can't, then we'll just get on with it. I'm really gonna make like an FAQ video, because there's a lot of questions that show up in the chat over and over and over and over and over and over and over again. There's a lot of overs for emphasis. Which I've answered a bazillion fucking times, and I'm really getting sick of it. So, yeah, I think I'm going to probably make an FAQ soon. Oh, thank you, Niles. Greatly appreciate it. Where can I buy these plants? Well, there's many stores in every city that you can buy plants. My entire apartment is full of plants. Like, I'm gonna just click over to the camera view for a second, just so you can see. It's hard to see. There's a lot of plants here. Like this is all plants. Like herbs down here. This is microgreens, and it's gonna be more herbs. This is greens up here. Behind me, there's a hanging passion fruit vine, and then on the other side of the camera there is just a shitload more plants. Like I'm going for like a tiny jungle thing. So. Yeah, that's been fun. So yeah, plants are wonderful, and it keeps it so nice and fresh in here. And I've been adding more and more flowers because I've got basically all the herbs and veg and stuff that I really want. So now I'm just kind of filling the rest of the spot with just pretty flowers that are. I try and pick flowers that are edible, just because I don't want to necessarily like fill the room with non-edible things, but certain ones aren't. Just because they're just pretty enough that it balances out. So, yeah, that's it. Yeah, somebody asked, Nausicaa inspired. I mean, a little. That's kind of my whole life, man. Like, that's the vibe I always go for. So actually, that reminds me. So one of the next, well, I don't know when it'll come out, but one of the videos that I'm working on is a Garduino project. It's Garden Arduino. So I've already started. I started wiring it up yesterday. If you follow me on Instagram, then you'll see the pictures. And So I'm basically going to be robotifying all of my plants, in the sense that like they're going to be watered automatically. So excuse me, it's gonna measure the soil moisture, and once the soil gets too dry, it's just gonna automatically water it, which is gonna be really great. I'm gonna be mixing Bacillus thuringiensis, BT. It's this type of bacteria that kills insects, because I have a bit of a fungus gnat problem. So, gonna need to rectify that. But yeah, so that's gonna get mixed into the watering solution. So like as it waters, it also kills off all the insects, so I don't have any of that problem, which is gonna be wonderful. Because holy crap, am I sick of frickin' fungus gnats. Um, but yeah, so be sure to check out my Instagram. You'll see all those updates. Very cool. Okay, back to the thing and the stuffs. Okay, so we need octanol synthase, octanol synthesis E. coli. So let's see. I think this is the one I was thinking of, but we'll see. Somebody was asking about how hot does it get in here. Before I had the air conditioner installed, it was quite warm. Like I had to like vent my apartment in the evenings just to deal with all the heat buildup. But now that I've got the air conditioner on, it makes no difference. Like it's actually it's too cold. I wish I'd turned that off before I started. Um, how are your pepper plants doing? Dead. I had to throw them out. They were definitely infected. I thought they'd gotten better, but they had not. So yeah, those, you can get an F in chat from my pepper plants. Okay, I think this is the thing I was thinking of, and also, see, this is what I love about metabolic stuff, and by love I mean hate, but also love. It's fascinating how freaking weird this is. So you got to figure that like like every time you see one of these compounds in this diagram, every single one of them, there's at least one enzyme that does that step. So like from acetyl-CoA to acetoacetate, like there's two enzymes here. So you got to figure that each one of those is a gene, which means that each one of those is expressed by the organism and has to be running and made, and it's got to be made in the right proportions, and it's a whole balance and dance. It's fucked. So by the looks of it, these are, yeah, okay, so this is interesting. So these are making different kind of alcohols, which, I mean, we're going for octanol. It's kind of what we want. Thank you for the F's in chat. So, you know, each one of these enzymes, so it's host, genotype, are talking about, like, different enzymes. This is not the paper that I was thinking of, though. So this is butanol, isobutanol, hexanol. Ooh, hexanol. So hexanol we did. Okay, yeah. So these are the now that I'm parsing this, each one of these is a gene, and you can see that not all genes are named nicely. Some of them are just strings of letters. But so by the looks of it, so hexanol, we did it in 4. This one looks like it's in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So this is 11 genes, which is obnoxious. So fatty alcohols is what happens when you get into like the really chonky alcohols. So like linoleic alcohol, I guess. Wouldn't be an acid, but you know, when you're at like 10, 11, 12 carbons long, then you're into like the fatty alcohols, because they're so large that they're like lipids at that point. They're oils. Okay, so this is not the paper that I want, but it's close. High production of fatty alcohols in E. coli. That's what I like to see. Maybe this is the one that does it in 4 genes or 5 genes. Okay, let's see. Background, don't care. Okay, 1, 2, 3, 4, 5, 6. Interesting. Okay. Somebody asked, how far is the Spider Silk project? When do you expect it could come out? I'm stuck waiting on reagents. I'm probably gonna, I planned on doing an update video this month, but my box of materials is currently sitting in Indiana. So until that gets, so it was shipped, and then FedEx lost it for a bit, and then it got shipped back to the person who shipped it to me, and so now I gotta wait on FedEx. Like I gotta wait for the shipment to get reshipped, and then get over the border, and then I can work on the spider silk thing again. So it's coming slowly, but you know, yeah. I think I'm gonna probably do an update video this month, regardless. I don't want to do update videos on stuff that there's not like a fun update. You know what I mean? Like the, yeah. Okay. So is this where the fuck yeah, this is still not the right thing. I think there was a paper that did it. Yeah. Okay. Yeah, 1-octanol has fuel characteristics similar to that of diesel. A synthetic 1-octanol pathway was engineered using octanoic acid generating fatty acid ACP thioesterase. Wow, that is a mouthful. Okay, deletion of ACRA reduced the synthesis of... Okay, that's fine. Boopity boop 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 boop. Okay, let's see what we got. Is there anything that just shows a frickin' pathway? Okay, there we go. Much better. So this is 1, 2, 3... 4, 5, 6. No, that's 5. I can count. This looks like 5 enzymes, I think. Yeah, that, I mean, shows 5. So this starts from glucose. There's this first enzyme that makes octanoyl ACP. This one makes octanoate. Then CARA goes octanol, which is, you can see that it's hard to see. I can't really zoom this in. But there's you can see there's a, or if you can, there's a double bond here. So the very last step is it just kind of yeets the double bond, and you get the alcohol thing on the end. So this looks very, very doable. So let's just have a little look. So yeah, so it says this work. So this is the results. This is telling you all the different ways that people have done this. So in this case, they used 1, 2, 3, 4, 5, 6, 7, just a shitload of genes, an absolute shitload of genes, and they only got a very tiny amount of octanol. So this is milligrams per liter. So, you know, not a huge amount, but like if you were growing this on huge scale, you could distill out a fair amount of octanol. And you got to consider this is turning from sugar. So the amount of sugar in versus the amount of octanol out is not super high, but you're also got to feed the organism. It's got to use the sugars for other things as well. Yeah, okay. So compared to all of this garbage that requires just an enormous number of genes to get this to work, this work does it in like 4, which is pretty wild. And they get a very reasonable amount of octanol out. So in this case, it says yield of octanol per unit of glucose. So in this case, they get 12 milligrams per gram of glucose. Whereas, you know, this much more complicated pathway gets 25. So it's about half the yield, but the amount of genes are way lower, so it's much easier to make. Also, because it's less genes, less pieces need to be working. And it's always easier when you're engineering something to use less genes. The more genes you have, the slower everything goes, because the organism has to make more enzymes to get the product that you want, and that vastly makes the system slower. So in this case, they're getting 4 milligrams per hour per liter, whereas this much more complicated system is getting 2.1 milligrams. So basically, what this translates to just summarize, you can get more octanol using this system, because even though you're getting less octanol per gram of glucose, you're getting it much faster. So in 14 hours, you get this yield, versus in 48 hours, you get this yield. So it's like, you can just grow more of it. Like, you can just grow it in a larger batch, and you'll get much more stuff. So in this case, they're growing this on M9. M9 is what's called a minimal media, so it's not like lysogeny broth, where you just basically burst a bunch of yeast and just feed the bacteria on like yeast goop. M9 is basically like salt with some sugar in it. Like, it's minimal. It's got the absolute bare minimum for growing bacteria. So that's that. So we got to get TS, SFP, CAR, and HAR to do the thing. I think I'm probably going to do the 2A peptide separator thing because I really like that system. And yeah, it's good. Um, and also, then it saves us on, whatchamacallit, promoters. And, finding more promoters is always a pain in the ass. So it's easier to just use one if you can. And so if we're going to do this in yeast, which I think we will, that could be really interesting because yeast produce much higher levels of protein compared to something like E. coli because they are physically larger. So they have more machinery that lets them make more protein, and their metabolic needs are different, but they can grow very large amounts of protein. And they're already you already grow yeast on basically sugar, so they already have a lot of the mechanisms for doing this. So it shouldn't be too much of a big deal to just shift. And also, you got to figure that part of yeast's metabolism is they make alcohols, and so this is an alcohol. It's a larger alcohol than ethanol, but it is an alcohol nonetheless. Okay, what else we got? Okay. Okay, so let's see if we can... I need the actual names of these enzymes, ideally. If I can find it. Okay, so... it starts with E. coli acyl ACP thioesterase. So it's called TESA, which is poorly named. Then it's carboxy. Okay, so these are the names of the enzymes. So we've got acyl ACP thioesterase, carboxylic acid reductase, fuck it, I can't even pronounce that. Transferase, and then AHR. It just it just doesn't say what it is. So these actually come from different organisms. So the first one's from E. coli. The second one is from M. marinarum, or marinum. What is this? M. marinum. Oh, Mycobacteria. Okay. Mycobacterium is a free-living bacteria. Opportunistic infections. Interesting. Mostly causes disease in fish. Okay. This is what I love about bio, where you can take these enzymes, which would never exist in one organism, and then just schmoo them together, and they all do the thing. Because they don't care about what organism they're in. They're indifferent. They just do the chemistry that they do. B. subtilis is another really common bacteria that people will engineer. So this is this is from three different bacteria that they take all these enzymes and shmoo them all together, and it works. So that's really cool. So we need the E. coli version of acyl ACP thioesterase. So we're going to copy that. We're going to paste that, and we're going to look up UniProt. So what did they call it? Called it TESA. Let's try that. TESA E. coli UniProt. TESA, thioesterase protease thing. Acyl-CoA thioesterase 1. That sounds more like what we want. It's from E. coli, acyl-CoA thioesterase. Acyl ACP thioesterase. I don't know what the difference is. It's the complete sequence. Where did this come from? If I click this, what happens? Oh, nothing helpful. Okay, well, let's go back. Yeah, okay. Did I just skip this? No, okay, it's right here. So I think the reason that it goes glucose to octanol ACP is because it basically just opens the glucose up. And glucose is actually a fairly long molecule. It's a ring, so if you open it up, you end up with a fairly long molecule. In fact, glucose, I can spell. Images. So yeah, here we go. So you can see there's actually, it's a fairly long carbon chain when you open up this ring, right? So you're already at a fairly long thing, and you're pretty close. So this is why they went with this route. Also, glucose is sort of like the universal fuel. So this is close. I don't know if this is the right thing though. Tess A gene. Why does it say acyl CoA? I need acyl ACP. I don't know what the difference is. What is this ACP? Okay, let's look this up and just see. Palmityl acyl carrier protein, ACP. Theoesterase. Okay. Palmitoyl acyl carrier protein theoesterase chloroplastase. Okay. What's this do? That's very weird. That's very, very weird. Actually, you know what? While we're on here, let's just look up TESS A. And then just narrow it down. See what that does. Oh, nothing helpful. TESS A E. coli. It's called TESS A. K12. This looks, I mean, this looks right, but I'm not sure. I mean, it says it's a theoesterase, so it should work. Acyl CoA, acyl CoA. This is always the hardest part. What did I just knock over? Oh. Balls. Unplugged my mic by accident. My bad. I really wish that it... Here, okay, let's... Ooh, there we go. Okay, so there's... So I'm gonna... let's see this. I really just want to find out what this is. Because I don't want to grab the wrong I mean, not that I actually intend on making any of this, but I would definitely not want to grab the wrong protein. Because then this just won't work. So I'm trying to, you know, teach good habits of, if you're going to do a thing, do a thing. In most organisms, this process... Okay. Fatty acids. So this is like a fatty acid processing thing. So they're kind of like... stealing a little bit of fatty acid processing, which is weird. Dozens of acyl ACPTEs have been functionally characterized. Fat A, Fat B, okay. What else do we got? Okay. That's plant. From Castor. Could that have just been a typo? Okay, what are the other references to this? In this study, we engineered a pathway that utilizes precursors from the natural process of fatty acid synthesis. Okay. Octanoic acid precursor was generated by incorporating a fatty acid, acyl ACP theoesterase, which was previously shown to have preference for 6 to 8 carbons. It says previously shown, so there should be a reference to this somewhere. Fatty acid. There's only 4 references to this. This is really frustrating. I wonder if there's a supplemental. So this is kind of where like stuff gets difficult. So I'm not seeing supplemental. I'm only seeing this one figure, which is really annoying. Huh. So they call it TESA, so let's try looking that up. Since the chain length of fatty alcohol was determined by the substrate specificity of TESA, this enzyme was therefore replaced with one that had been previously shown to generate an 8-carbon fatty acid. Oh, here we go. This is the paper. Didn't we just open this? I feel like we just opened this. Yeah, it is. That's weird. Oh, shit. TESA. What? Are you kidding me? Okay, so just to catch everybody up, because this is very frustrating. We've found this paper that says you need this enzyme from apparently E. coli to do this, except I can't seem to find any frickin' reference to this thing anywhere. And it's like, well, this was determined in this paper. Okay, so we go to this paper, and where the fuck is the thing? Results. Okay, that's a thing. So this is just the phylogeny of all of the things that do this process. Okay. Coli. The in vivo substrate specificities of acyl ACP TEs were determined in E. coli. Based on their specificities, these enzymes were clustered into 3 classes. Act primarily on 14 and 60 carbons, broad substrate specificities towards 8 and 14 carbons. So, okay, so basically there's a whole family of these enzymes that all work, that will all basically spit out different length carbon chains. So each, these ones, some do 14 and 16 long, some do 8, some do less, some do more. Okay. K27, 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 yeah, that's not helpful. Expressing a given acyl ACP, okay, fine. Yeah, this phylogen, excuse me. Ooh, hello. Somebody, I might have found something. Uniprot POADA1. Uniprot POADA1, I think. P0 ADA1. Yeah, see, this is the one I was looking at before. TESA is multifunctional esterase that can act as a thioesterase, lysophosphatase, and protease. Okay. Palmityl-CoA. What's this from? Oh, this is from E. coli. Hexadecanol CoA. Interesting. Pretty small. Okay. We're going to go with this one, because I'm sick of looking for this, and this is close enough. But, I mean, it seems like this is an enzyme which is fairly reactive, and will make all kinds of interesting weird... Oh, see? All ACP. So this does work on the right substrate, I think. I'm seeing at least a couple ACPs, so it should do the thing. I think. Hexadecanol. Yeah, I don't know. Okay, cool. All right, we're going with this one because I'm sick of searching, and we need to start somewhere and move the show along because I got other things to do. So we're gonna just copy that. Bio.ru. So we did say that we were gonna do yeast this time. Paste to paste. So we're gonna do Saccharomyces cerevisiae because that's what this works in. Okay, so what I just did there. Oh, shit. Or what I am doing here is this string of letters here, which I got from Uniprot, which was here. Uniprot is sort of like a database of proteins and enzymes. So I think this is the one that I ended up using. Yeah, P0ADA1. Um, this is the first enzyme in the pathway that makes octanol, in theory. I could have the wrong one, but unfortunately, see, this is why like anytime somebody's like and like scientists write papers and therefore they're professionals. Yeah, but then they write papers like this where they're referencing a thing that doesn't exist and there's no supplemental where they list the sequences. Having written a paper does not make you a good scientist necessarily. It makes you good at getting published, I suppose, but like having papers that are poorly written like this where, they reference a random name for a thing and they don't give the exact like enzyme number. Like a lot of the time these enzymes come with identification numbers. It's this EC number a lot of the time. So if you're not listing this or giving a reference or the sequence or whatever to the enzymes that you're using, you're just an asshole. Like, what is the point of writing your paper if it's not replicatable? The whole point of science is you're supposed to be able to replicate the fucking thing that you publish. If you're just doing it for cred and to like put it on your resume, what is the fucking point? Oh wow, hello, Mason. Thank you very much, man, or lady. I don't know what you are. Thank you very much, random internet person. Greatly, greatly, greatly appreciate it. Okay, back to what we were doing. So yeah, if you're just to end my rant really quickly here, if you write a paper, include as much information as you physically can. Like if it would have taken the people who wrote this a fraction of a second to include an identifier number next to each of these, and it would have made my life a bazillion times easier. So yeah, fucking be professionals. Don't just be assholes who write papers. Anyway, rant over. So anyway, we found this enzyme which we think is probably the right one. This is the amino acid sequence of the enzyme. So we copied this, went over to this site, pasted it in, and now we're telling this site what we want it to do. So we want it to translate this into DNA, and we want it to do so. The exact DNA usage is a little bit different between organisms. So if we were to choose like human, so Homo sapien, it would give us a different DNA sequence than if we choose Saccharomyces cerevisiae, which is baker's yeast. Even if we chose Pichia, it would give us a different thing. And the reason is the DNA is broken into 3-letter codons, and you can actually see them here. So M, see, like the first letter's an M. This is a methionine. So there's different things that code for, well, actually there's only one thing that codes for methionine, it's ATG. But for some of the other ones, for example, let's see if I can find one that's got a nice double in it. Yeah, okay, so you can see this here. So I is, I believe, isoleucine. The codon, so the 3-letter DNA code that codes for isoleucine, can either be ATT, ATA, or ATC. And you'll see that there's a percentage number here. The percentage is basically what percent of the time, or what ratio of the time that organism uses this specific combination of 3 letters to mean that it wants this amino acid. So if you only use ATC, for example, you're going to get lower yield because the organism doesn't use this code as much. So you got to consider that all organisms are physical systems. So when we say doesn't use this code, there's actually another chemical that does the transfer of the amino acids to the growing protein as it's being synthesized. And so there is literally lower levels of the tRNA. They're called tRNA or transfer RNA. There's literally less of them that code for ATC than there is for ATT. So if you're so this program is basically going through this chart and picking so like, for example, this is L, I believe this is leucine. So it mostly uses a T and then a T, and it very rarely uses C. So if you use TTC, it will technically make a leucine, but the yield will be god awful because there just isn't a lot of tRNA that can like do that flip. It mostly uses the other ones. So when you're choosing organisms, that's what it's choosing. Anyway, so now it's going to translate this for me from amino acids into DNA, which we can work with. So let's click reverse translate. Wonderful. I'm going to just grab the DNA sequence, copy, copy. Now we're going to go over to here. So this is the plasmid that we made in the very, very first stream. And if you look at it, you can see that there's a couple interesting things in here. The thing that I've got highlighted is ovalbumin, which is another word for egg white. Excuse me. So egg white is primarily ovalbumin. It's like 50% ovalbumin. The rest is mostly water. So this plasmid is built to make egg whites in yeast. So this is a plasmid that works in yeast. It's not properly labeled because fucking nothing ever seems to be. But in here, there's a chunk in here and there's a chunk over here that matches a sequence on the actual Baker yeast genome. So like these overlap. The bit that's between them is going to be different. But if you take this piece of DNA and you cut it with a specific enzyme, and I want to say it's NTOI, but don't quote me on that because I don't actually know. It should cut right here-ish and right here-ish. And so you end up with like a linear fragment of DNA, like basically something like that. And then when you put this into yeast, the organism recognizes that there's a damaged piece of DNA, and so it just like tries to find a match. And so what it'll do is it'll recognize that this chunk over here matches a spot on the yeast genome, and so it'll basically shove this whole chunk into the yeast genome, and then this becomes a permanent part of its genome, so that this whole gene system lasts much, much longer. So yeah, that's this. So what we're going to do is we're going to get rid of the egg white, and we're also going to get rid of this piece. This is alpha factor secretion signal. Alpha made is the other name for this is alpha mating factor, and basically this is what's called a secretion signal. So this is actually protein, like it makes protein. This chunk of protein here comes from a protein which is always excreted by the yeast. So as long as this tag is here, the yeast recognize anything that's stuck to it as this needs to leave the cell. So you can use this as a way to take things that you make and have it just be excreted, which in this case we don't want. So we're going to get rid of that, and that's annoying. I hate when it does that. So we're going to get rid of the secretion signal, and we're going to get rid of the ovalbumin, and so we can just go delete, bye-bye. And then we are immediately, without touching anything, wait, shit. That's not right. That's not right at all. I forgot. We can't do that. There's supposed to be another frickin' restriction site in here. I put it in here. Where did it go? Or is it up here? It's up here. So we can do this. I stand corrected. BAMHI. Okay. I need to just do a couple of little checks. So when I designed this plasmid, I designed it such that it had these various restriction sites in here. The restriction sites are spots on the DNA, which if you mix this DNA with this specific enzyme, it's literally called BAMHI in this case. The enzyme will cut the DNA at exactly this spot, and it'll cut it in this way. So you see how the line kind of goes like, whoop. That's literally like, so you'll end up with a dangle on the cut DNA. It's called a sticky end, whereas this one, small one, you'll see it cuts straight down, so it's called a blunt end. So you've always got to cut with things that you know are going to cut in the exact spot that you want. So in this case, we're going to do BAMHI because, nope, BAMHI has 2 cut sites. So we're going to do SMI, which should have 2 cut sites. One should be here, and then the other one should be here. Yeah, okay, that's... So yeah, I added, when I had this made, I had small 1 or SMAI sites added to this plasmid to make sure that if I cut with small 1, which is an enzyme I have, it'll leave blunt ends. So what I'm going to do is I'm going to go from here to here. Nope, come on, fucker. So that should be highlighted at the cut site. No, it isn't. Okay, so I click on the enzyme that I want to cut with. So that's going to highlight at like it's going to put the marker at exactly the cut site. Oh, thank you. Is your name Bollocks? Well, thank you, Bollocks. You made interesting username choices. Anyway, so if I cut with from small one and I hold shift and I click on here, it'll highlight from cut site to cut site. So now if I click delete, it should, it derped out. Where did it go? Why does it always do that? Okay, so you'll see that there's still a small one here. Because, you know, the front half and the back half match. So, like, this is now exactly where we can put our DNA. So, let's do exactly that. So, I'm going to click paste. This is the bit of DNA that we just reverse translated. I'm immediately, before I click off on anything, I'm going to name it. So, it was Tess A from E. coli. We're going to click Save Annotation. I also just want to change the color briefly, not for any particular reason, but just because it's easier visually to see what's going on. So now we've got our first protein in here. Wonderful. I'm just going to nip over to Cheeseberry McPearface because there's a piece of DNA that I want. Specifically, it's called a 2A peptide. 2A peptides are what are called self-cleavable linkers. So we've used them time and time again on the streams. I use them all the time. They're fantastic. So basically what they do is as this DNA, or like, okay, so basically what happens is, let's stay here. So this bit of DNA starts being read and turned into RNA. It's going to actually make RNA all the way from there to here. So you end up with this really long stretch of an RNA copy of this DNA. This is called transcription. When that RNA is then read by a cluster of enzymes called a ribosome, it will start turning that RNA into protein, which is what we want. As it's reading the RNA, and you know, sticking each one of these amino acids on, eventually it hits the 2A peptide, which is here. So somewhere in this peptide, it basically makes the enzyme skip, sort of like literally think of like a record skipping, which I realize is a bit of a dated reference, but I mean, it's the most accurate thing I can think of. So it literally makes the thing skip and let go of the protein that it was making, and then it just immediately keeps going. So you end up with like two, or in this case, 1, 2, 3, 4, 5, like 6 or 7 proteins all off of a single piece of RNA. So this is very efficient. It doesn't do the skip every single time. So you do end up with weird fusions part of the time, but that should be fine. So we're going to just copy the 2A peptide. There's a whole family of these peptides. They come from a virus. I don't remember which, but they are viral in origin. So we're going to just copy the 2A peptide, and then we're going to go back over to... Shit, I just realized what I did. Balls. I needed to do something else. I gotta undo this really quick. This will just take a second. I need to basically undo all the way back to the base state, because I started working directly in this Plasmid, and I can't damage this file. So come on, keep going, please. There we go. Okay, so this is now unbroken. So I need to go to the actual folder that this is in. I skipped a step. I just started working. That was dumb. Octanol. Octanol? Octanol. We're going to go with Octanol. So now I've got a new folder. We're just going to go back. This is just some like organizational stuff, and also you'd never mess with your files once you've made them, or you're going to be very, very sad. So I was just using this directly. That was really dumb. So I'm going to copy this to that new folder really quickly, Octanol. And then we're going to just redo that again. So it's the exact steps that we just did. It's just non-destructive edits are very, very, very important. Because otherwise you end up with sadness. and nobody wants that. Okay, so I'm going to close the original one so I don't accidentally fuck it up again. So let's just do that same thing again. So we grab from small 1 to the other small one, which should be at the bottom, which is right here. Click, delete. And now before we move anything, I'm just going to recopy this, which was over here. Copy. Okay, I'm literally just doing the same steps that we did a second ago. I'm just trying to not wreck them. Where did it go? Where's the cursor? What the fuck? Oh, that's the Terminator. Ooh. Panicked for a second there. Okay, paste that in. Cool. And now again, we're going to create our annotation. Test A, E. coli. I'm also going to, so it's P0A DA1. POA DA1, yeah. Okay. Cool. And I'm going to give it a color. We went green last time, so we're going to be consistent. Now we're going to go back to Cheeseberry McPearface. Again, grab the 2A peptide. Yep. Now we're going to go to the end of this and just go paste. So now we've got our 2A peptide. Wonderful. We got 1 enzyme down. We got 3 to go. That took a little bit of doing, but we got there eventually. Okay. Back to our thing. Okay. Now we're looking for carboxylic reductase from M. marinarum. Very different kind of marinara. Carboxylic acid reductase. Yeah, this. Search. Mycobacterium marinarum. This looks like the right thing. So we're going to grab this. I think this is the right... Oh my god, that is a big protein. Okay. It's a chunky boy. So yeah, you can see that not all enzymes are tiny. Some are gigantic. It's sort of just the way things play out. But I don't actually intend on getting this synthesized, so I don't really care. So we're going to do the same thing that we did before, which is we found the enzyme. We're going to reverse translate it. We're going to copy it. Copy. Wait a second. Is that... I'm going to just add a 0 to this just to make sure that it's all on one line. Copy. Cool. Now we're going to go back over to here. And that could go there. Before I do anything, I'm just going to rename this. Octanol. Wonderful. Interesting. Okay. Okay, now I'm going to paste that in. I don't know why that thinks that that's a 2A peptide when it's definitely not. This is carboxylic acid reductase. I need to adjust this because this is not right. What's the position here? Base number 10977. So just go here. Oh, shit, what? 2140, okay. There we go. Fix that. So now we've got 2 done. I'm going to just copy our 2A peptide here. Wow, that is a humongous protein. Holy crap. We're going to just stick this here. Right on the end. Got our 2A peptide again, which is wonderful. It's interesting, there's a small one side here. I'm just having a quick look at the comments just to see what people are talking about because I haven't looked in a little bit. Alright, bits and bobs. Okay, cool. So we're 2 down. We got 2 to go, and then we should have Octanol, which would be neat. And what's fun is you can then take this and add the ATF1 gene that we looked at the very beginning of this, and it should smell like oranges, which is kind of cool. Okay, moving on. So we got these 2. We need B subtilis, phosphopantethional transferase. Okay. From B subtilis. Here we go. That was easy. I like it when these ones are easy and they're not just like, and then you need to find a thing from a paper and then do a thing. So sequence status complete. That's definitely what we want. FASTA. We're doing the same thing as before. We're just working our way through it. So we've got the sequence. We're going to do the reverse translate, pasty paste, reverse translate, copy. I'm just working quick because we're just burning our way through this. We're going to paste that in. We're going to then immediately name it. I don't remember what the fucking name was, so we're going to just copy that, go over here, paste this in. B subtilis. Just want to make that note. I'm going to change the color because I like making sure that things are colorful, just because then it's easier to see what's going on. We're going to grab the 2A peptide again. Now, it's actually really bad practice to do this the way that I'm doing this, where I'm using the same 2A peptide. There's P2A, there's C2A, there's a whole bunch of different ones. It's best to use different ones rather than the same one over and over again. But because I don't really care about this plasmid, we're just kind of burning our way through it. I'm just going with it. Rex asks, basically, the long and short of this question is, how applicable is benchling to doing work with a company? Depends on the company. I know lots of people who use benchling. It really depends what you're doing. What was the question? Inquire asked something, and the response was very snippy. Apples says, how did you ever find the octanol specific FAT1B gene? I didn't. Is that next? I don't know. I hadn't gotten that far. Yeah, this doesn't use FAT1B. It uses FAS. In theory, which is not listed here. Very strange. From FAS. Fatty acid. Okay, that's not helpful. There's only 2 references to it here, but I think that one is in the. I think this is already in the enzyme. Like this is already in the organism. Not sure. Okay, we're on the last one. It says E. coli AHR. Not even gonna bother to give us the name of the fucking enzyme because these people are lazy as shit. Yeah, you can see my regard for scientists doing their job poorly is not high. I have no idea if this is the right thing because it's not fucking labeled, so I have no idea what it's supposed to be. Joseph, oh my god, there's 7,000 thing. No. Okay, let's. Oh, for fuck's sake. This is what I mean. This is like they're professional scientists who can't be fucking bothered to like label their shit properly. And it drives me absolutely up the wall. Like, please beat these people over the head with a pipette until they learn how to not suck at their job. And yeah, I will say that about professionals, because what kind of professional doesn't fucking label their thing properly? AHR, reduce, okay, that's not helpful. It just says aldehyde reductase. Okay? Which one? Fuck's sake. And AHR, an aldehyde reductase, previously known as... Okay, see, why wouldn't you just put that in the fucking... Okay. Here we go. AHR E. coli. I mean, this is what I mean. Like, you could have just saved me the time if you just fucking write your papers properly. Christ. Alright, that's not what I want. Okay, wait. Not what I wanted. I wanted the FASTA sequence from this. Okay, FASTA. And I do believe that after this one, we're done. Pasty paste. Annotation. Reductase from E. coli, but I'm going to make sure that I labeled this properly. P27, because that one was actually annoying. And we're going to make it red for no particular reason whatsoever. Okay, cool. So that should be all of them. I just need to make sure there's not a stop code on here, because I have this sneaking suspicion that there might be a stop code on here. There is not. Wonderful. Okay, I always get worried about that, because if you end up with a stop code on somewhere you don't want it, that can be a huge pain in the ass. 1 Hour Eclipse asks, why is this in my recommended? Because maybe you like science. Come hang out. We're doing science. I don't know. There's a bunch of other people in here who seem to enjoy themselves. So yeah, welcome. Just expand that a little bit. I just want to scroll over. I don't know why it won't let me. There we go. There's the scroll. Okay, just seeing what's going on. Cool. So that should actually be the last of them. So we've got our TF1, 1, 2, 3, 4. Okay, I think that's all of them. I think we're done. I think we are actually done. That's wild. So let's just throw a stop code on here, and then call it a day. Forward translation. Okay, TAA should do it. Wonderful. I like doing 2 or 3. Forward. Cool. So what I just did there was I just added stop codons. So this is basically the final end of the protein, basically. So with that done, let's just do a review of what we've done. Is there a reason it's called octanol synthesis instead of octanol? Because I'm shit at spelling. That's the only... And look, I fixed it. Yay! Okay. Just quickly, Riley asks, if I wanted to complete any of your projects from your GitHub, what are the necessary steps to produce these in real life as someone with not much lab equipment? Okay, so basically what you'd want to do is you would start with getting like the same backbone that I used. So whenever I start doing the editing, I'll always say that, like, and we cut with this enzyme. So in this case, it was small one, which that site has been destroyed. But if we open up, where the fuck was it? So I'm going to go over to the milk plasmid just so I can show you this. So in this case, this milk or this egg plasmid will eventually be made available, so you could buy it. So what you would do is you would literally buy this piece of DNA from probably AdGene, because that's where I intend on posting it. And then you would have to get this. So actually, you wouldn't have to get shit. You would just tell the company to do this. So what you would do is, if you wanted to make this octanol synthesis plasmid, what you would do is you get this piece of DNA from probably AdGene, and then you send it to a gene synthesis company, and there are many. And the specifics of that vary depending on the company. Some have like an online thing where you can do all your submissions. Sometimes you just have to send them an email, whatever the case may be. You get a quote. So you'd send them basically you send them this, and you tell them in detailed instructions what you want them to do. So you say, I need you to print this. So you literally, like, in a Word. So the company that I use likes emails and Word documents. So this is what I do. So I would copy all of this code, literally like the letters, and I stick it in a Word document. And in that document, I would say, print this sequence. And so they would literally, like, print out this bit of DNA. And then you would say, take this plasmid, cut it with this enzyme, and then put the two together. And from there, you could then say, make sure that you make a version of, like isolate a clone. So then, okay, I'm skipping a step. So you'd say, basically, cut with this enzyme, put in this piece of DNA to make this plasmid. And then I also include the entire sequence. So I'll literally go like Control A, select the whole thing, and then stick that whole sequence into the Word document with it. And I'll also highlight the sequence that they should have inserted, just to make sure this is as clear as physically possible. And then I'll say, take this piece of DNA, put it into some E. coli, and then isolate a clone which has exactly this sequence in this direction. So I would say, like, this sequence needs to become immediately after this part in this order. Because if they flip it upside down, the whole thing won't work. So you say, isolate a clone that does this, and then send me a mini prep. So that would be, you grow that once you find an E. coli that has successfully taken in this completed bit of DNA, in this order, in exactly the way that you've ordered, you say, take that clone, make a bazillion more of them, like literally grow like 100 milliliters worth of media with this E. coli in it, and tell them what the selection agent is. In this case, this would be a yeast, and you'd be like, make sure that it's AMP resistant or whatever. Or no, actually no, this would be E. coli and then AMP resistant. Anyway, it doesn't matter. Point is, you'd say, find me a clone that is the perfect finished sequence, that's exactly what I ordered, grow up a lot of it, extract DNA from that batch of E. coli, and then send me however many tubes you want of that DNA, and send me what's called a stab of that E. coli, so that I can also do the isolation if I want. And then you send them that order, they'll give you a quote, and they'll tell you how much that'll cost. In this case, this chunk is huge. So I would say, holy shit. Yeah, this would cost on the order of, I would say, actually, let me check. How large was this? Because this was quite large as well. From here to here. So yeah, okay. So for context, this piece is 4,294 base pairs long, and this cost about $1,200 to have made. So octanol synthesis, which is a little longer, actually I can shorten that down a little bit, which is about 6,000 base pairs long, I would estimate would be in the range of $1,500 to $1,800. So they would say, my face freezed? What? What the hell? That's weird. Why did my camera freeze? What the fuck? Alright, give me a second, y'all. I'll sort this. Very weird. Add source. Integrated camera. What the hell? I have no idea why that just happened. That's new and horrifying. Alright, well, we're just going to ignore that for a second. I'm going to just finish this up, and we're going to call it for the day. Yeah, I can't unplug it. It's built into my laptop. Okay, well, that's really weird. I don't know what the deal is. Just for those wondering, I've already ordered some upgrades. I'm literally waiting on one cable. So in future, I'm not going to be using my laptop camera. I'm going to be using a proper camera. I just needed a power cable for it to make sure that it doesn't die midstream. So yeah, that's really frustrating. But as long as you guys can hear me, my stupid face doesn't really matter. So yeah, let's just wrap this up. So the point is, you would tell them all of these things, and then they would give you a quote, and then you give them money, and you send them the piece of DNA that you want to stick the backbone into, or the insert into, and then they would do this. And so you'd say, cut with small one, insert this, put these two together, all that kind of stuff. So yeah, that's that. But yeah, so let's just review what this does. This plasmid has 1, 2, 3, 4 different genes in it. Each one slowly builds up towards Octanol, which is a medium-length semi-fatty alcohol. In this case, I can show it to you because there's a picture of it. This is not a good picture. Okay, well, whatever. This is the pathway that we just set up, basically. So we've got TES-A, we've got CAR, we got SFP, and we've got AHR. Those are the different things that we've got in here. So these all come together to produce a compound called Octanol, which is very similar to diesel. So basically the way this would work is you would put this whole plasmid into yeast. This is built to work in regular normal baker's yeast. And then once it's in the yeast will take in sugar and then turn that sugar into Octanol. Most likely this will get trapped in the yeast, so what you'd have to do is grow very large quantities of yeast, and then basically treat it kind of like Vegemite or Marmite production, where you save the actual, like, cake of yeast. Oh hey, the camera unfroze. For no obvious reason. Okay, whatever, fuck it, we're going with it. So anyway, you treat it basically like Vegemite, where you get like a cake of yeast, and then you would pop them all open and essentially distill out the octanol, and you would end up with biofuel. So that's really cool. So that's the thing. That's the whole pathway. So like all the other plasmids, this will get uploaded to GitHub. I think I'm still missing one or two because I'm a turnip and I forget these things. But every plasmid that we've made on the streams will eventually, at the very least, be added to the GitHub. GitHub. This is the GitHub for those who are interested. So yeah, these are all the different plasmids that we've made in previous videos. You're free to download them, do with them what you please. I'd recommend checking it out. Seems like there's a fair number of people watching it now, which is really cool, because I can't code for shit, but I can do DNA code. But yeah, so that's the plan. This is the thing. This will be uploaded. This was fun. We'll be doing. We won't do another Who's Gene next week. Next week is going to be. Here, I'm going to just set this to camera because it's done. Just camera. Is this working now? No, it is not. Why isn't this working? What the hell? I'm just fixing this. Give me a second. Any of these work? Hey, this one works. Okay, cool. Just fixing this. Give me just a moment. Okay, we're good. We're back. Okay, fixed. Cool. So I'm gonna just kind of do the wrap up, and then I'm gonna take a little bit of time to answer some questions, and then we're gonna call it for the day because it has almost been 2 hours. So thing number one, the plasmids, as I said, are gonna be on the GitHub. Definitely check that out. If you haven't already, then be sure to check out the new video that I just posted on my second channel on the Taste Emporium, and in fact, I'm gonna pull that up just for those who are interested. Just pulling this up. Okay, so I'm gonna just switch this back to scene for a second. Cool. So this is my second channel. For those of you who have not seen this already, it's called the Taste Emporium. Isn't it lovely? This is the new video that I just put out. I'd highly recommend checking it out. It is legitimately one of the prettiest things I've ever made, both food-wise and video-wise. I'm in contact with a couple professional chefs, and I'm working on some interesting collaboration stuff with them, and that's going to be super fucking exciting. I'm thrilled about this. They're like Michelin-starred quality chefs, and it's so cool to be able to interact with them and you know, exchange ideas and stuff with them. But yeah, you know, I got some really great feedback about this, and you know, they were thrilled. So highly recommend. Yeah, I am subscribed to my own channel. It was, yeah. But yeah, so anyways, be sure to check this channel out. Highly recommend. It was a really good video, I think. I'm quite satisfied with the way it turned out. I spent a lot of time on it. It was like, I was up at like 6 in the morning making dacquoise on the the day that it went out, just because I was like rushing to get it done. But it is the way that it is. Okay. But yeah, so those are the updates. I'd highly recommend checking those things out. There were some other updates at the beginning, but we'll talk about that as they come, essentially. So yeah, I'm going to take a little bit of time to answer some questions, and then we're going to call it for the day. And then we'll be back on, what the fuck day is it? It's today Friday. We'll be back on Monday with a biohack chat with my friend Gabe. If you have suggestions for topics that you want us to talk about, then send them to me on Twitter. It would be very nice to be on Twitter and see something pleasant other than the absolute clusterfuck fire that is the rest of the planet at the moment. So, video ideas. Okay, let's answer some questions, and then we'll call it for the day. Vivek asks, which software do you use for editing videos? I use Adobe Premiere. It's kind of the one I've gotten used to. It has its issues, but this is part of the reason we're building a new editing computer. Do you use Anki to memorize bio stuff? I don't know what Anki is. Yeah, so no. Have you considered doing a basics video for those who are new to the subject? Yes, I'm starting to work on it. I'm just kind of collecting my thoughts and what things I want to include and that sort of thing. But I am starting to work on that, so that'll be out eventually. Have you considered asking some PC channels for help with the PC? No, because I can put a PC together just fine. I've done it many times. Do you follow other genetic engineering YouTube channels? As far as I know, it's not that they don't exist, there's just not that many, and I have some interpersonal issues with some of them. Like, Josiah technically has a YouTube channel, but it'll be a very cold day in hell before I ever support anything that he ever does. Just fuck him. Somebody asks, are you building the computer yourself? Yes, that is the plan. And technically you save money if you build it yourself. Technically, but it kind of let me put more points into the components. Is the best way to put this. Um, and yeah, so I'm actually going to be doing a caseless computer. So I'm going to make like a wall panel essentially with just everything mounted on it, and might do some lights just because why not? Got to get that aesthetic. But yeah, that's gonna be a fun build. I don't know if I'm gonna do a video about building the computer. I might. I just don't think it's gonna be particularly interesting because there's approximately a billion other videos on how to do this. So yeah, that's the thing. Is it actually legal to design such yeast and let it be made as an average EU citizen? Design, yes. Make, no. You're allowed to design whatever you want. Like, there's no laws against using benchling and designing DNA. You're just not allowed to make anything in the EU without, like, crazy amounts of licensing and bureaucratic bullshit. So I have some EU friends who are very good genetic places like the US actually do the genetic engineering because it's legal there. So it's this like really interesting international collaboration, which I think is really cool. And I like doing it. We do all kinds of fun stuff together. How much do you pay for the grow lights, and are the blue-purple lights really better, cheaper, more effective, or overrated? I think technically you save a little bit on energy costs with the blue and red lights, but honestly, I don't think it's worth it. Like, all the lights I've got behind me are the equivalent of one 120-watt light bulb. And that's growing, that's 3 shelves worth of grow lights. And they're just, like, cheap shit, 4000K, like, intensity, like 20 watt per strip LED fluorescent style lights, and they work really well. The plants are super happy, so I'm fine with it. And again, it's the equivalent of one 120 watt light bulb between all of them, so that's great. The other grow light I have is a 3500K like light temperature grow light. It's a 1000 watt. I don't actually know if it's 1000 watt or if it's 1000 watt equivalent. It's very damn bright though, so that's the thing. I don't like the blue and red LED grow lights because they hurt my eyes. It's an obnoxious color and I hate everything being pink. I only buy full spectrum grow lights because I want it to be nice and feel like the sun in here, not like I'm in some fucking like it already feels enough like it's a spaceship in here. I don't need it to actually like be also blue and red. It's hard on my eyes. I don't like it. Okay. Moving on. Inquire asks, what about getting it from the outside already engineered? Again, you're gonna have huge problems with that. Like, owning genetically modified organisms in the EU is like a whole headache. If you're in the EU, you're sort of shit out of luck, is fortunately the best I can say. Their laws are archaic and stupid, and written by people who don't know their ass from a hole in the ground, so that's sort of the way it is. I mean, you can do some stuff, but like it's weird. Okay. Ibrahim asks, how do you do this radiation-eating bacteria that could make nuclear waste safer? So eating the radiation wouldn't make the nuclear waste safer. It would just be a way to use the waste as a fuel source. Like the radiation, like if you have nuclear waste and you stick it in a hole and then you fill it with 200 feet of concrete, like the radiation isn't getting out. It's more of just like you can use that radiation rather than it doing anything. The only way that you're going to get rid of that radiation is to burn the nuclear material, and I don't mean in fire, I mean burn it in a fast reactor. And that's a whole conversation for another day that isn't really on topic. Would you recommend The Molecular Biology of the Cell by Alberts? Well, I haven't read it, so no. Probably is fine, but I'm not familiar with it. Favorite most useful enzyme. These are two different things. My favorite enzyme is any of the luciferases, just because they're magical to me. I love glowing stuff. I always have. Not particularly useful though. It's pretty, but in terms of useful enzymes, that's hard to say. There's a lot of useful ones. I'm more of a structural protein kind of guy. I have a tendency towards silk, but meh. What else we got? won't it leak into the ground? That is not how radiation works, and that's also not how nuclear waste works. They don't just like dig an actual hole and just like put it in. It's all in like sealed containers, in like multiple layers of containers, and it's embedded in concrete. And you only do nuclear disposal in areas that have no aquifers, are geologically stable, and like they've found really great places to store nuclear waste where you can store it forever and no one will ever touch it. Except people always bitch when you go to do this because like it's like they can't, they don't want you to like, they don't really want you to have nuclear waste or dispose of it. And also it's always like sacred sites or whatever. I'm like, it's a fucking mountain. Just nuclear power is to me more valuable than somebody saying that this mountain is sacred. Fucking not my thing. Yeah, like a world powered by nuclear means we don't go extinct. To me, that has more value than somebody's belief system about a fucking mountain. But I understand that I'm biased, and you know, these things are important to some people. Um, fucking. Yeah. Anyways, distracted by a fungus gnat there. Who said nuclear waste can't be sacred? I mean, it is to me. It's a magical rock. It grants death to anyone who sees the blue glow. Yeah, fast neutron reactors are endless power. Yeah, basically, like you get, okay, so in just because I've struck on this chord and I feel like ranting about it. In a traditional nuclear reactor, you get about 1% of the energy out of the fuel before it's spent. 1%. It's already the most efficient technology on Earth and is the only truly carbon negative, potentially, system, because it is the greenest technology by a fucking mile because of the sheer energy density. It is the safest technology on Earth, and yet that's only getting 1% of the energy out of the fuel. In a fast reactor, you can get at the bare minimum 30% of the energy out of the fuel, if not up to 90 to 100%. Like it's a huge difference in energy recovery. All of our nuclear waste is not waste. It's still fuel. It's just we don't have the right reactors to use it as fuel. If we used it in a fast reactor, you could burn it down, and instead of it being radioactive functionally forever, you can burn it down to basically cesium, where it'll be radioactive for like a couple hundred years instead of a couple 100,000. So it gets rid of all the plutonium, it gets rid of all the transuranic elements. It is, there's definitely some concerns because you do end up with a much more enriched amount of certain elements, but it is fan-fucking-tastic. It's great tech. I think in the right hands, it could be fantastic. Even if it was only like certain countries had the fast reactors and everyone else just had slow reactors, being able to use the spent fuel and recover energy from it and then use that energy to power carbon capture or any of the other things that we need, because energy is a requirement for everything we do. If we actually had this, then we would be in way better situation than we currently are. Nuclear is fantastic, big fan. One person died from Fukushima. Were they standing next to it? Like, more people die from coal. Like, more people die from radiation from coal every year than have ever died from every nuclear accident combined. What are your thoughts on ITER? I think it's a very expensive donut. I don't know. It's neat. I'd like to see it work, but I also think it's meh. Yeah, anyways. Shit. Okay, Hiroshima and Nagasaki are not nuclear powered. Fucking, there's a difference between nuclear bombs and nuclear power. Making a nuclear bomb is very hard. Thing number one. And thing number two, they decay very quickly, so you constantly have to be, like, upkeeping them. And also, a nuclear reactor can't blow up like that. That's not how it works. This is one of the many reasons why I find that new Chernobyl show on HBO to be absolute ass garbage. Like, they fucked up the science so badly. Like, it's like more fear-mongering bullshit in a time when we need nuclear power. It's so fucking irritating. Um, anyway, I digress. Um, yeah, in theory, ITER will be. So the thing with ITER is it's very expensive, but it's not just about building the reactor. It's building the facilities that make the reactor parts. So once you've got one ITER, it's very expensive to make one. But once you've got one, it's much cheaper to make many more. Do you have the means to make a nuclear bomb? No, I fucking... Do I look like a nation state? Fuck off. Like... Anyway. Do we need an entire... We need an entire garden filled with biohacked plants. I think it would be really great. I agree. I really wish that the use of genetically modified plants was way more acceptable. And I wish I could have a glowing garden full of plants that do all kinds of interesting things. That'd be great. As a nuclear supporter, I still wouldn't say it's 100% needed. Connected grids of renewable... Okay, no. Fucking no. Solar and wind are useless. It takes more carbon to produce a solar panel or a fucking wind turbine than they will ever produce the equivalent amount of energy in their entire lifespan. Like, solar panels wear out very quickly. Unsurprisingly, a thing that's exposed to radiation all the time wears out very quickly. It takes more carbon to make one solar panel than that solar panel will ever generate, like the energy equivalent that it will generate in its entire fucking lifetime. Doesn't make sense. Nuclear is the only system that actually produces more energy than you put in to process the ore, by like a mile. And again, that's on 1% efficiency, not the 40% or 50% that we could have with different reactor designs. And rent. Yeah, this is like most renewables are like, I like the idea of renewable. I don't think it's actually gonna solve shit. Unless it's nuclear. Nuclear is the only truly like green tech. All the rest are just like PR, essentially. Doesn't make any sense. Why don't we fund new nuclear reactor designs? Because people are easily scared, and you have scaremongering like, say, the new Chernobyl show, that scares people away from nuclear. Even if you add up all of the nuclear accidents that have ever happened, more people have died by installing solar panels. And yeah, it's the thing. Also, if you've ever seen a wind turbine, like, destructively fail, it's quite a spectacle. Like, they really go kaboom. Like, they get going too fast and they just like spew bits of fan in every direction. Like, more people have died from windmills than have died from nuclear. And that's like, you got to consider that's nuclear. That's like the worst nuclear accident, which was Chernobyl, doesn't hold a candle to most of the things that have happened. Also, if you go to like Pripyat and like the area near the like Chernobyl exclusion zone, it's like full of life. All the animals have come back and stuff. It's wonderful. Well, like, wait a second. So they go off and it keeps the humans out of nature for a little bit. I'm sorry, this seems like a win to me. And also, yeah, Chernobyl was only as bad as it was because they were trying to cover it up more than they were trying to actually deal with it. Like, if they'd actually distributed the potassium pills, like the potassium iodide pills, immediately and actually not tried to, be hella Soviet about it, like, it would have been fine. But they did, so here we go. Okay. Yeah, it was also a terrible reactor design. Like, there were flaws with it. There were huge flaws with it. And like, there were known flaws with it before it blew up. Whereas a modern new style reactor, you could literally fly a plane into it and it wouldn't explode. They're designed that way for a fucking reason. All the reactors that have gone kaboom were very poorly designed, and every time a reactor fails, we learn from it and we make every new reactor more safe. Even we retrofit all the reactors that are currently working. There are reactors that are the same style as Chernobyl but are way safer because they've fixed the issues with it. That's the thing. Yeah, hydro is kind of green, except that the amount of energy that goes into building all the concrete and upkeep of the dam itself is still carbon positive, is the problem. Yeah, Fukushima was also, like, it was just peak stupid. Like, they had the backup generators in a basement in a tsunami zone on an earthquake. Like, everything kind of went wrong. And it still only kind of went, like, poof. So yeah, that's the thing. Okay. The main point against nuclear is not the risk, but the nuclear waste. Okay, let me explain something to you. 40 years of nuclear waste fits in something like 10 55-gallon barrels. Like, it's the size of, like, a couple large trucks. It's 40 years of waste off of one reactor. And it's always stored near the reactor in, like, a little fenced-off, like, highly secure warehouse type thing, usually. Also, again, nuclear waste. Well, actually waste. It's just fuel we haven't decided to use yet. Like, there's this misconception that nuclear produces all this horrible fuel. Okay, the thing that most people are worried about is the waste that comes out of the core. The waste that comes out of the core is highly radioactive. It is quite dangerous if you don't know what you're doing, except everyone who handles it knows what they're doing. Also, tiny amounts of it. Most of the nuclear waste that's produced globally is not from reactors. It's actually medical waste, and used in radiation therapy and that sort of thing. It's mostly like plastic bags and stuff that have been contaminated with radioisotopes that are used in medical imaging. Those isotopes decay down to nothing in like 5 or 10 years. So you keep them in a warehouse, you don't touch them for 5 years, and then they're safe. You can put them in a normal dumpster. It's fine. So that's the thing. Like, most nuclear, like, all of the world's global nuclear waste from every reactor on Earth can fit in like one small building. Like, one proper nuclear waste disposal site, like a deep mine. Think like a deep old salt mine kind of thing. Like, one mine like that could hold all of the world's nuclear waste for something like 1,000 years worth of nuclear waste safely, before you'd fill it and you'd need a new one. This is the thing. There is no argument against nuclear power. It doesn't exist. There are no good arguments against nuclear power. Nuclear power is fantastic. Anyway, what about all the leaks from nuclear waste transportation? It's not a thing. Like, it's massively stupid has to happen before that happens. The people who handle nuclear materials are they're regulated globally. Like, it's a huge international collaboration, and they take this shit very seriously because it's nuclear waste. If you fuck it up, you die. The more dangerous something is, the more careful people tend to be when handling it. Nuclear waste is no exception. So they don't just kind of like handle it willy-nilly. Yeah, the only argument is it's scary. That's not an argument. Like, anyway, thoughts on fusion? I love fusion. Fusion sounds great. It's going to be 30 to the end years before we finally get it, but big fan. Okay. What else we got? Yeah, I mean, fusion, like, they keep making progress on it. The new Stellarbrators look really cool. I mean, even ITER, when it's finally built, could actually produce proper amounts of energy. Tokamaks are interesting because they are a very easy way of getting the energy back out. Like, in a lot of fusion systems, like, you can produce enough radiation. Somebody just kicked something in. Where did that just come from? Oh, thank you, Straff. Fuck. Okay, so fusion is interesting, especially tokamak. So Canada almost had a functioning tokamak, in the sense that not functioning in like it turns on, functioning in like it made power. The only reason that we don't is literally because of bureaucracy. Like, Hydro-Quebec had a working tokamak that was like built for power generation. It is now sitting in the fucking Ottawa Science and Tech Museum. I've seen it, and it kills me that it's sitting there because we were like this close. Like, Canada is one of the only countries that is not involved in ITER because our fusion tech is actually like fascinating, and we're really damn good at it. What the fuck is the name of it? It's called Global Fusion. There's a company in BC that's doing Uh, Easily the coolest fusion reactor that I've ever seen. It's got these big pistons on it, and so it's a big sphere with all these huge pistons on the outside. Um, oh, thank you, Lawrence. Greatly appreciated. Um, so yeah, it's got these big pistons on the outside. On the inside, it's a sphere of spinning liquid metal with a void in the middle. So they're basically like pumping the liquid metal through this void, um, or through this chamber to like, so, and they're spinning it fast enough, just through pumping. Like they pull the liquid out at the top and the bottom, and they inject it in the sides, and so it is this liquid shell of liquid metal, that's spinning around, and then they inject extremely hot plasma into a void in the middle. And if the timing is just perfect, they use the pistons to then push on the liquid and squeeze the plasma physically, not with magnets, with big fucking pistons, and they just like squeeze the whole thing down such that it's a fusion engine. Like think about like the compression cycle in a regular engine. Yeah, it's like a tokamak, but done with pistons. But yeah, think about the compression cycle in a regular combustion engine. You squeeze the fuel down, it goes kaboom, and it pushes everything else. So the heat is directly transferred, and all the radiation is directly absorbed by the spinning liquid metal. The heat is then piped away, like the liquid metal is piped away, and the heat is transferred directly to a heat exchanger to run a turbine. Like, they literally are making a fusion engine. It is so fucking cool. And I give it about 15, 20 years till they get it done. But, like, they've been working on this a while, and they're getting close, and it is dope as hell, and the Canadian government is investing heavily into this. This is one of the reasons why Canada is one of the only countries not invested in ITER, because we think our shit works better. So, yeah. Very cool. It'll never be in a car. It's a huge machine. But it can run a city. So that's pretty cool. Okay. Let's see what else we got. What are the risks of DNA synthesis machines becoming as affordable as... So it's... The machines themselves are cheap shit. The reagents for DNA synthesis are where it's expensive and highly regulated. So actually getting the reagents is hard. Because they don't want every random schmuck just being able to print whatever DNA they want. Oh, thank you, Clarissa. I'm getting a Ryzen 9, I think. I'll get you in fact, I'll pull that up really quick. I have a whole list of, oh, shit. What happened? So yeah, I'm going to pull up I've got my list of computer parts. So I'm just pulling that up really quickly. Give me like 2 seconds. Crap, where did it go? Okay, yeah. It's the AMD Ryzen 9 3900X. Is the CPU that I'm going for. Super excited. Okay, we could see microwave Bluetooth. Can we see the lower end of infrared, but our ballot? So we actually can see into the near infrared, technically. So a friend of mine, Gabe, you've met Gabe, he's on the biohack chats, did an experiment. It was actually a recreation of an earlier experiment, but him and this guy named Jeff did this experiment where they basically ate a special version of Soylent, which was completely deficient in vitamin A. So there's actually two types of vitamin A1 and vitamin A2, and your body is fine to use either, but it vastly prefers vitamin A1. If there's any vitamin A1 in your body, it will use vitamin A1. If you're completely deficient in it, though, you'll use vitamin A2. Vitamin A, regardless of the type, is used in your eyeballs. It's retinol. It's used in your vision system. So if you completely deprive your body of vitamin A and then give it vitamin A2, which is ridiculously expensive, which is why that they did the deficiency rather than just trying to supplement it really hard, then your eyeballs will actually incorporate vitamin A2, and it shifts your vision over a little bit. So instead of blue to red, it shifts it over a bit, so your blues look kind of yellow, which is weird, but you can also see into the near infrared, and it works really quite well. Wouldn't suggest anyone try this because depriving your body of vitamin A for extended periods of time is not good for you, unsurprisingly. Yeah, so that's that thing. You can. There's also other ways to do it. There's what are called up conversion nanoparticles or down conversion up conversion nanoparticles, where you can take two infrared photons and like shmoo them together into one photon that's like green, for example. And so it would look to you like green light, but you're actually seeing infrared light. And they've done this in rats where they made these up conversion nanoparticles and then they injected into their eyes with a protein coating that makes it stick to the cone cells, or rod cells, I don't remember which. Yeah, it works really well. And then the rats were able to see infrared, which is fucking cool. So yes, technically doable, very hard. I wouldn't test it on myself because I don't inject things into my eyeballs because I'm not a fucking idiot. Can you see the body heat? No, that is very deep infrared. Like, that's not near infrared at all. Near infrared is kind of weird. It's what your phone can, or what certain phones can see, but it's not like heat. Hydrogen boron fusion is theoretically great, but actually getting it to work is very hard. Like, the laser that you use to run that thing is fucking massive, and like the amount of energy, it's like, okay, great, you can do hydrogen boron fusion, you can use a big fucking laser to do it, but how do you get the energy back out? Like, technically on paper there's more energy coming out than you're putting in, but how do you capture it? That's the problem with most of these fusion systems. It's technically possible to get that like energy output, but how the hell do you capture it in a useful manner is what's hard. This is why the general fusion concept and tokamaks are so popular, because the energy capture is so easy. So yeah, they're technically difficult to build, but if you can do them, the energy like harvesting is easy. Which graphic card am I planning on buying? I am planning on getting a Asus GeForce GTX 1660. At some point, I'm thinking I'll probably upgrade it to something better, but I figured I'm getting kind of like the bare minimum to do like VR stuff, basically. But eventually I'll upgrade it to maybe like a 2080 Ti or something. But I mean, those are freaking expensive, so I'm like, I don't really need it for this right now. And besides, a 1660 is already much better than the graphics card in the laptop I currently use for editing, so I think it's a big step up regardless. Okay, what the fuck, Mr. Maguma? What's Mr. Maguma saying? Nothing. Lucas asks, how do you know all of this stuff? What are you studying? Great videos. Well, thank you. I'm glad you like the videos. How do I know all of this stuff? I read a lot. I also got my start not in biology, but in nuclear physics. I was building myself a nuclear fusion reactor when I was 14. Everyone needs a hobby. Which I was doing so that I could use the neutron radiation that comes out of it to irradiate copper, because copper, when irradiated, forms copper-64, I believe, which decays via B+ decay, which is antimatter. So I was building a fusion reactor so that I could say that I made a little bit of antimatter in my basement. Everybody needs a hobby. I studied biology, though, but I dropped out before I finished the degree. But that's the only actual, like, official stuff that I took. Like, I only did a couple of years. I did 3 years of a bio degree without finishing it, and that's my only official schooling. Everything else is self-taught. Are there any new physics projects you're working on? Holograms. I've been meaning to do holograms for a very long time. My helium neon laser just arrived. I'm waiting on, in theory, it was supposed to be delivered today, my piece of optical breadboard, which I'll be using to set the hologram stuff up on. I'm waiting on my hologram plates, which should arrive in a week or two. And then, so I'm going to be basically using Ben from Applied Sciences' method to copy holograms, like physically. Because I was actually working on that before he put his video out, but he managed to figure it out before I did. So I'm going to give his method a try, just to show that it works. I'm also going to show how making holograms works, how copying holograms works, the different types of holograms, how holography works. I've got all kinds of really cool stuff planned, and I think it's going to be a lot of fun. So it's going to be a big video. So I'm just waiting on supplies at this point. Is an ATP engine possible? Kind of, probably. I don't know. Maybe. I can't give you a better answer than that. Oh, thank you, Garan. Seeing, wait, what? I love your neonic resistance bees convo. Oh, thank you. I'm glad you like that. What is the future of energy storage tech? Supercaps, ideally. I'm a big fan of the stuff that Robert Murray Smith is doing, but I think it'll be a little bit till his batteries ever actually see the light of production, just because it's a huge amount of investment to actually make a product. But I mean, if he can get it working, his graphene-based batteries are dope as hell. How far are you with the glowing plants? So I think we needed 4 genes synthesized for the glowing plants, and 2 of them have been successfully synthesized. 2 of them failed in synthesis, so they're now being sent out to a different company to try and make them. And if that fails again, then we're gonna have to use a different company. But it's progress. It's bio. It's slow. What do you think about Elon Musk? I think he's a rich asshole who, like, people pray at the altar of Musk for absolutely no reason. Like, dude's like trying to force his workers to go back to work when there's a fucking pandemic because reasons, and then he's throwing a hissy fit to get the city to kowtow to him. I'm not a big fan. I like SpaceX. SpaceX is fantastic, but he didn't make SpaceX. The engineers made SpaceX, and the engineers are awesome. I have big respect for the people who work at those companies. I have very little respect for the people who head those companies most of the time. There are exceptions. Tory Bruno, the guy who runs ULA, is fantastic. Big fan. Whereas Musk, bit of a cult following, not into it. Do you find that people who finished a degree look down on you? Some of them do, but then science circles around them and they can kindly go fuck themselves. So I don't really care. Also, it's rare these days. Like, when I was first getting started, like, people, like, I've had experiences where people who have degrees definitely, like, didn't even want to give me the time of day because I didn't have, like, there was this one person that I knew who, the first time I talked with them, I was, they did a bunch of, they were like a nanotech researcher, and I was like, hey, I've worked on this stuff and I'm interested in this, and, trying to ask them questions. They didn't even care. They didn't give a fuck what I had to say. Their only question was, what is your degree? Where did you do your school? I'm like, that shouldn't matter. I'm asking you a technical question correctly. Just answer my fucking question. Let's just have a normal conversation. Why do you need to? And because when I said that, I'm like, I went to this school, but I didn't finish my degree. Literally just went like, dead, and just literally like turned away and just stopped talking to me. I'm like, are you kidding me? So yeah, but that hasn't happened in a really long time. These days, most people give me like, I mean, I have a whole portfolio of all the things that I've done. I don't think anyone really thinks that I'm just some schmuck anymore. Anyway. Can I please know why you grow Mimosa pudica? Mimosa pudica is known as the sensitive plant, and so when you touch it, the whole plant contracts. And you can actually get electrical signals off of it when you do this, and I'm going to try and pick them up and then pick those signals up with an Arduino using another amplifier, and then use that to like trigger electronics, because I want to like boop a plant and then have it like turn my lights off. That's it. That's the whole reason. Okay. What else we got? Yeah, the when I was in Brazil, there, like, everywhere you go, like, Mimosa pudica just grew everywhere. So like you'd be walking through a field and the whole field was like wilting out of your way. It was very weird and kind of magical. I love that plant, which is kind of why I wanted to grow it. It's a special place in my heart. I love it. It's such a neat plant. But also I've got a plan for it, so it'll be fun. Shit. I unplugged my freaking mic again. Ow. Gotta stop doing that. Keep kicking that. All right. Well, it is almost 4:15. So I'm gonna do like one more question and then I'm gonna wrap it up for the day. No, I unplugged the power to the mic, so that can like mess with audio levels and stuff. Um, okay, let's do one more question and then Derek, I'm not even fucking justifying that with an answer. That's fuck off. um, is there any kind of exchange for propagated biohacked plants? I mean, you can ask the people who made them. Most people are pretty good. Oh, thank you, Swed. Swede? I can't pronounce your name, but thank you very much. Greatly appreciate it. Is there any exchange for propagated biohacked plants? You got to ask the people who made them. Honestly, there's no official exchange that I'm aware of. Okay, this one I want to do, and then I'll probably Swed. Okay, well, thank you, Swed. Greatly appreciate it. Oh yeah, so what will the next food video be? So the next food video is going to be a recipe from Shokugeki no Soma, which is one of my favorite anime, because I'm a gigantic weeb. That is a millefeuille with mushrooms and ants. So I get to cook with some insects, which will be fun. So yeah, that's gonna be a lot of fun. I've been practicing my millefeuille. If you go on Instagram and you look up Taste Emporium, there's a picture of the first millefeuille that I ever made. It was a vanilla custard with strawberries, and I made every single part myself. And so the new one that I'm going to be making for the video is a millefeuille with mushrooms. So it's a duxelle of mushrooms on the inside, and then a shiitake confit on top, and then it also uses ants in the duxelle to add like a tang. But then there's also like some sauces that go with it, which I've been thinking about, because they show it in the show, but they don't actually talk about what the sauces are. So I was thinking of doing I wasn't quite like I forget the name of it, but I was thinking of doing like maybe a fond de veau type sauce on the side, but there were 3 different colors of sauce, so they could be different. I don't know. I'm gonna figure it out. Ought to be fun. Ants or ant larvae? Ants. Like here, wait, I can. Are they? Oh, I'm gonna grab. Give me a second. Ants. Yeah, culinary grade black ants. So yeah, that's gonna be fun. Yeah, so the reason that these are used is because they contain formic acid, which, like citric acid, add like a acidic tang, but it's not citrusy in the way that citric acid is, or that we're used to. It's apparently different. I haven't tried these yet. You can see the wrapping still on it. But yeah, so it's a duxelle of mushrooms and shallots and cream and ants. The puff pastry, shiitake confit, and then slices of truffles on top. So I bought like a super cheap, tiny little jar of truffle slices just so I could like put them on top. And then, but yeah, that little thing was like $25, I think, which compared to buying a whole truffle is nothing, but it's enough for the video. Also, I'm not the kind of nutsack to actually buy a whole truffle. Those things are fucking expensive. Isn't it toxic? Formic acid? Yes, in large quantities, but in the quantities in ants, it's so tiny. It's teeny amounts of formic acid. It's not enough to cause a problem. Like, eating ants is well known. Like, it's a thing. They're culinary grade. These are meant for food. So as long as you're not if you ate that whole jar, you'd probably be pretty sick. But I'm going to be making like an infusion of ants, probably in some brandy. Um, so I'm going to just get the formic acid out, and then just add small amounts. Um, yeah, so in small amounts, formic acid is fine. In large amounts, formic acid is not fine. So just enough to give it a tang, not enough to be a problem. And yeah. Also, like, tongue is quite sensitive to acidity, so you're able to taste it without it being at toxic levels. Yeah, I mean, and like poison is a dose thing. Like water, apples are poisonous if you eat enough of them. Like if you ate like a handful of apple seeds, you would die, just from cyanide poisoning. So like, it's fine. Um, have I ever made black garlic? No, I've never made black garlic. I've cooked with black garlic before. But making black garlic, you got to basically keep a thing at 60 degrees for a month, which is a little bit obnoxious. Slow RAR. I don't know what slow RAR is, but thank you, Bollocks, for your notification or your donation, and also interesting choice of username. um, how do you spell the food so I can find what it looks like? You know what? I'll just pull up a picture really quick. Give me like 2 seconds, and then I can just show you. Images. This is the thing. So this is the millefeuille. So you can see it's got the shiitake confit on top, a duxelle in the middle, and so you see these different sauces. These are not referenced in either the manga or the anime, so I'm gonna have to make these up. I'm thinking of like a mushroom reduction sauce thing, like a brown sauce. I'm blanking on what that's called, but that's what I'm thinking. Um, but yeah, so you can see the truffle slices. I'm not fucking buying a whole truffle. That's ridiculous. But the shiitake confit is quite easy to do. The duxelle is quite easy to do. Puff pastry is a little tricky, but it's going to be an excuse to do it. There's also other recipes from Shokugeki that I want to do, but this will be the next one. So yeah, it's going to be this one, and then after that, I'm going to be doing double gluten bread, not gluten free, double gluten, from Yakitate, Japan. Um, because I've been experimenting with adding extra gluten to bread, and it is fucking magical. So yeah, that's gonna be fun. Um, Also, I've noticed the people who don't like mushrooms have just not had good mushrooms. There's a lot of mushrooms. Many of them are vastly more delicious than the kinds that you usually get in the grocery store, and also mushrooms need to be cooked properly. Um, yeah, so most people who don't like mushrooms are just have not had good much. It's the same way like most of the people who are picky with the foods that they eat, I'm like, you just haven't had good food. Like I have a friend who can't really like she doesn't like eating meat, not because meat is bad, but just she had some like bad experiences with it when she was younger. And like every time I've made something for her, she's like, you know, this is delicious. I'm like, no shit, you just gotta cook it right. um but yeah, so that's um yeah. More than Canadian flour. No, I mean I have a bag of it's called Vital Gluten. It's like it's gluten powder. So I added like a tablespoon to the last batch of buns that I made, and they were fucking magical. It was awesome. Highly recommend. Um, you describe yourself as a happy geek. I have no idea what that means. I mean, I'm happy. I'm a geek. I suppose so. Anyways, I'm gonna have to wrap it up for the day at this point. Thank you everybody who has donated. Really, truly, greatly appreciated. There'll be more. There'll be more videos coming up. There's the THC yeast video next week on Saturday. I will get you the exact. It's either at 2 or 4. I don't remember which, but it's around that time. if you've enjoyed, you know all the things to do. Like, subscribe, all that shit. Um, but yeah, there just quickly, there's some other videos that I've got like slowly burning in the background that like once they're ready are going to be just absolutely fantastic. I'm waiting on some supplies. More videos will be coming out this month. So yeah, stay tuned. Be sure to tune back in. There'll be a biohack chat on Monday. If you've got ideas for things you want me and Gabe to talk about, send them to me on Twitter. And yeah, that's where I'll leave it for today. I hope you guys have a good day. Stay safe. If you're going to be going out, wear a mask. Be careful. World's very scary right now. Just be smart. Yeah, and have a good day. Be awesome. Bye.